Well, thanks, Paul. And yeah, welcome everyone. So thanks for the introduction. Justin and we will tag team this presentation. Let me see if I can drive my screen share. How's that looking? Good. Okay. So good morning, good afternoon, good evening, everybody, wherever you happen to be in the world. As Bo pointed out, it's Wednesday morning here in New Zealand. The sun is shining. It's a wonderful day. Thank you all for tuning in to the webinar. And of course, a very big thank you to the Tree Fund as well for giving us this opportunity to share the research with you. This is something Justin and I completed around about a year ago. Things being what they are, this was just published just 2 or 3 weeks ago. So as we get to the end of the slideshow, we'll share some information with you about how you can access the publication. Whoops. Okay, so we're going to begin with just a little bit of background. Nothing too crazy. We just want to sort of cover off some very basic concepts that will sort of underpin the research elements of the talk. Then we'll talk to you about the research questions, the methods that we used to investigate those questions, the results of the experiment, and then we'll leave you with something that is hopefully practical and useful to you. And for those of you that work with roots, perhaps something that you can use immediately if you felt so inclined. Okay, so we are talking about roots today, but we're going to go to the top of the tree to begin with, and we're going to look at some branches, and we're going to look at some different ways of pruning branches. So if we look at this slide on the left-hand side, we can see this sort of graphic of a topping cut or a stub cut, whatever you wish to call it. And the typical response that we would see there, of course, is this kind of whirl of epicormic shoots emerging from the top. And then over time, we may start to see some discoloration or some decay emerging in that central column. On the right-hand side, the two photographs there, this is what that might look like in practice. These are a couple of trees not too far from where I live that I just photographed quite recently, freshly made stub cuts. So no epicormic response on these just yet, but we might expect to see that over time. Now, there are some nuanced circumstances when we might think about leaving stubs on trees, but ordinarily, this isn't normally the way that we would prune the branches of a tree. This is probably a more typical way of doing things. So if we look at the birch tree on the left-hand side, this birch tree is having a haircut. So we're going to remove the red piece at the end and reduce back to the side branch, the little side branch there. Now, when we think about aspect ratio, which is the ratio of the diameter of the little side branch that we leave behind to the diameter of the parent branch that we're pruning, that was probably no more than 2 to 1. So I would expect this birch tree to respond quite well to this reduction cut. So that branch should assume a new terminal role quite successfully, and I would hope the parent tissue would wall off all the nasties at the pruning cut. On the right-hand side, this is what that might look like in practice. I'm sure everyone is very familiar with this kind of pruning work, but this is the more typical way that we would prune the branches of a tree to, I guess, make things shorter, as it were. So what we're trying to illustrate with this slide is that there are differences in compartmentalisation efficacy between seasons and between species. So what does that mean? Well, let's take an oak tree, for example. If we were to prune the branches of that oak tree, if we prune those branches in summer, maybe the oak tree's codit response is optimal or an optimized codit response, whereas if we prune those same branches in the winter, maybe the oak tree's codit response is suboptimal, or it's less than optimized. Whereas a tree of a different species, an ash or a beech, for example, may respond in the opposite or a slightly different manner. Now, there are some reasons for this, but they're not too important right now. But just keep this principle in mind as we go through the rest of the slides. So what does best practice tell us about pruning roots? So some of you perhaps have got a copy of this best practice document. This is the ISA's latest guidance notes on root management. And what the guidance tells us is that root pruning cuts should be made cleanly using a sharp tool appropriate for the root diameter, leaving a clean cut perpendicular to the direction of growth with the bark intact. So in practice, we would find the root, using some careful excavation techniques. We would expose that root carefully. And once we decided where we're going to make the cut, we'd use some kind of sharp cutting tool, make a nice clean cut at 90 degrees to the direction of growth, cover the root over with some soil perhaps, maybe moisten it off if things are sort of hot and dry. But after that, otherwise, sort of hope for the best. What the guidance doesn't tell us, at least just yet, is where and when to cut. So here are the research questions that Justin and I asked. Number one, do tree roots respond differently when they are severed in different locations, internodally or by making a reduction cut? So stubs or back to side roots. And the second one, do tree roots respond differently when they are severed before or after an active growing season, spring versus autumn, or for those of you joining us from America, the fall? Now, working with roots is an inherently dirty business. Anyone that has ever worked with roots before will know it is very dirty. Those of you that have an interest in the sort of the science of arboriculture, if you follow the research with roots and root pruning, you'll be aware that research over the last few years in this area has been rather scant. And I think it's just because there aren't too many people that want to do this. It's very mucky business. It's very time consuming and it can be quite unpleasant. So big thank you again to the Tree Fund for supporting Justin and I in our root pruning endeavours. The pictures you'll see on the screen there are from another Tree Fund project that we heard about earlier. It was my PhD program a number of years ago. And I've been operating an air spade for several days at this point, I think, looking for roots to include in my study. And as you'll appreciate, I am rather covered in filth. So anyone that's ever operated an air spade will be aware, of course, of how unpleasant this can be at times, not something that you would ordinarily want to spend too much time doing on your own. However, help is at hand. This is Justin, and at this point, I'm going to hand over to Justin to talk to you about the methods. I'll try to anticipate your slide timings, Justin, but just give me a nudge when you're ready to shift.
Perfect. Sounds good. So yeah, no, nice to be speaking to everybody today. I think it was sort of already explained, but I was Andrew's PhD supervisor a number of years ago, and we've maintained sort of a good working relationship over the years, despite living in different cities. And it's sort of our shared interest in tree roots that sort of continues to bring us back together. So I guess a few years ago, Andrew called me up and said, I'm proposing to work on a new project with you. And I said, I'm listening. And he said, it's about tree roots. And I said, go on. And he said, you get to work an air spade for almost a month continuously. And I said, I'm absolutely in. So I was excited to work on the project with them. And so we sort of developed the idea, and ultimately we convened in Christchurch to undertake this work. So Christchurch is a city in the South Island of New Zealand. Andrew's just going to activate the little graphic there. So yeah, South Island of New Zealand on the east coast. It's a city of roughly 400,000 people, and it's sort of anchored. The center of the city is this wonderful park called Hagley Park, and you can see Hagley Park taking up sort of the right half of your screen there. It's a very large sort of urban park, well maintained, has roughly 100 odd years, maybe 120 odd years of history. And the specific trees that we were working in there are just sort of bisecting that image from lower left to sort of upper right. Those are the trees that we worked with for this particular project. So those trees are somewhere between 80 and 100 years old. We're not entirely sure the age. They're plain trees. You can see from the images there, they're relatively large. Trees grow very quickly in this part of the world. And so even though they're only 100, maybe 80 to 100 years old, they're sort of 1.2 meters on average in diameter. And exhibit many of the characteristics that you would sort of expect from mature or veteran trees. So we worked with, as the slide shows, 32 of those trees over the course of our project. Now, what we undertook, or what we endeavored to undertake, was this implementation of these root pruning treatments. So Andrew's already sort of highlighted what the sort of the overall goal was, looking at the seasonality of the root pruning, as well as the position of the root pruning. So what you see in the images here is us implementing those root pruning treatments. So the first step was really to use the air spade to, I guess, identify and expose the roots that we were going to prune. The idea here was to be, I guess, as sensitive as possible to the trees. We didn't want to sort of expose more roots than we needed to. So we agreed to work roughly 3 times the DBH away from the trees, and essentially excavate a trench using the air spade until we were able to identify, I guess, 3 roots per tree. And we were looking for roots of reasonable size. Not too big, not too small. We'll tell you about the root characteristics in a little while, but yeah, ultimately we were looking for 3 roots per tree that we could prune. Now, what you can see there in the image at middle, oh, sorry, just back 1 second, Andrew. Yeah, what you can see in that image in the middle is sort of this white spray paint that we used to identify the roots that we were ultimately going to work with. Yep, next slide. So in this image, I'll sort of continue talking to you about the methods that we used. These are our root pruning treatments. At left, what you can see is the root equivalent of a stub cut, and that's what we called our internode treatment. The image in the middle is, again, the root equivalent of a reduction cut, so that's back to a side root, and that's what we called our junction treatment. And again, here we pruned half of these trees in spring and half in autumn or fall. And then we took those removed pieces of the root and we brought them back to the lab where we used that to work out the cross-sectional area of the root. Now, I guess if you're working with roots typically, or I suppose rather, if you're working with a circular shape, working out the cross-sectional area is easy. You can just sort of measure diameter, but of course, roots don't share that same sort of circularity. They're quite eccentric in shape. And so we had to use other methods, which I'll talk about a little bit later on to measure that cross-sectional area. Okay, so once we had pruned those roots, we had to basically map them, for want of a better word. We had to essentially identify where they were relative to the tree, so that when we went back later to sort of see how the root response had engaged, we'd be able to find them relatively easily. So we took lots of photos. At one stage, we considered basically just leaving some little metal objects in the soil at each of the roots and then use a metal detector to identify the location of the root pruning. But the urban forester for the city told us that actually there's a lot of people who use that park for essentially treasure hunting, for yeah, metal detecting, and so we avoided that. And yeah, we just basically took lots of images. We took lots of measurements to basically take a distance and bearing from the center of the tree to each of the pruned roots. And ultimately, we built up a map of where the root pruning had been undertaken, so that when we did return to those trees, we'd be able to find them relatively easily. Now, one of the points that I'll make is when we backfilled after the root pruning, we used sort of native soil from that site. So we didn't sort of use any sort of amended soil. It wasn't sort of a higher quality soil. It was just the native soil from that site that we backfilled into those holes. And once we'd done that backfilling, we waited and we waited and we waited and we waited. Originally, the application that we'd made to Tree Fund was it described a 2-year experiment, so we were going to wait for 2 years to see the root regrowth. Um, but I guess after 2 years, we just had a hunch that maybe we should wait an additional year, just to really sort of maximize the amount of root growth that we'd or root regrowth that we'd see. And so we wrote to Tree Fund and asked them whether they'd be comfortable with us extending the timeline for another year. They were happy to do so. So 3 years later, back we went. And this picture now shows Andrew on the airspace this time, working very happily to expose the roots that we had previously pruned 3 years after the fact. So once we'd identified the locations of those previously pruned roots, what we were able to do was to harvest those roots for further analysis. For some of the smaller roots, the things that are maybe roughly the size of a finger or a couple of fingers, we use loppers. So that was an effective way to get those root samples. For the larger roots, we began using a silky saw, but of course we dulled and destroyed, I think, 3 silkies within the first half day. So we abandoned that fairly quickly. And so the image you see at left is me holding a saber saw and cutting out those root samples. Now, what we tried to do was basically take a sample that was roughly 100 millimeters in length, so 10 centimeters. What's that? Roughly 4 inches, I guess, in length, back from where the original pruning cut had been made. Now, what the images at right show is basically, well, the one at center is showing the typical root regrowth response. Andrew and I have been referring to that as sort of a beard of root regrowth. So lots and lots of relatively small roots that are regrowing from the cut face or sort of surrounding the cut face, the calloused area around the cut face. And at right, that's a relatively unusual form of regrowth, so we thought we would highlight that one. We're referring to that as that sort of face sucker from the movie Alien. It's got just sort of 4 or 5 sort of relatively large roots regrowing from that callus tissue. So that's ultimately what we did 3 years after having implemented those initial root prunings. So then we eventually took those samples back to the lab. So again, 3 roots per tree times 36 trees, 32 trees, excuse me. We brought them back to the lab where we cleaned them up, washed them, removed all the dirt, ensured that they were well labeled, and that prepared us for the next extremely tedious step. So the next step was measuring all of that regrowth. And what you see at left there is Andrew undertaking that work. So I was taking the photo, sat beside him with a laptop to enter the data that he was basically measuring. What you're seeing is Andrew using a set of calipers to basically measure the diameter of each root, where that root was with respect to the pruning cut, and how many roots there were per original root. We measured, well, Andrew measured, let me rephrase, Andrew measured a couple thousand roots this way, and I entered the data as he was doing it. So luckily we had a sunny day, so we were able to do it outside. It made for, I guess, as pleasant an experience as possible. After all of this work, what we knew was how many roots, how big they were, and where they were with respect to the original pruning cut. What you see at right is a couple of examples of that. The regrowth from the original sort of callus tissue in that original root in the center image, and at right, the I guess the root after we'd sort of cut off all of the regrown roots as part of that measurement process. So a couple more photos. Some of the root regrowth responses that we looked at was essentially where the regrowth occurred. So the first thing I'll point out is that sort of rim of callus tissue where many of the roots are reforming or growing from. So that was a very common occurrence, something that we observed on virtually every new root. And then the quadrants that you see there, 1, 2, 3, and 4, is that's going to help us answer the question of where the roots were regrowing from. So we did I guess hypothesize that we might see asymmetric root regrowth. And we weren't entirely sure where that root regrowth would occur, but we thought we might see asymmetric root regrowth from different quadrants. And so you can see that quadrant 4 and quadrant 1 represent the top of the root, or the root that the surface of the root that was closest to the soil surface. And then quadrants 3 and 2 on that image represent I guess the half of the root that was lower in the ground. Once we had measured everything, all of those regrown roots, we dissected the root down that vertical axis. So from top to bottom using a bandsaw, so that we could subsequently, I guess, measure discoloration or I guess a proxy for decay. And so what you see there at left, the image at left, is basically that bisected root where we cut it down the middle using that bandsaw, and we were able to, I guess, describe that area of discoloration again as a proxy for decay. We are aware that decay and discoloration are not synonymous, but we called that a decay index in any case, and we described that by this calculation here. So the area of discoloration in square centimeters divided by the cross-sectional area of the root in square centimeters. And that gave us an indication of how much discoloration, again, a symptom of decay, had occurred in those roots 3 years after pruning. So by the time we'd done all of this, all of our measurements, what we had was a variety of different root responses to those treatments. And again, just a reminder, those treatments were autumn or spring pruning, and those treatments were also internode or junction root pruning cuts. So that is pruning back to a side branch or stub cut. The responses that we had were the number of new roots, the position of those new roots on the parent root, the diameter of those new roots, and the sum of the cross-sectional area of regrowth as a percentage of the parent root. And then finally, a decay index, a measure of discoloration. Now, this is one that I'm quite excited to share with you. This is not something we did for every one of the trees. In fact, it's something that we did for only the first tree. Originally, Andrew and I had intended to measure root regrowth length. That was something that rather excited us. But on day 1, we began sort of exhuming the root regrowth, and we started following the regrowth with the air spade, and we were following along, anticipating that it would terminate, but it just kept going and going and going, and ultimately we were successful in getting that one bit of root regrowth out of the ground. But you can see that it extended for nearly 2 meters, 1.87 meters. It grew on average 1.7 millimeters per day over the course of those 3 years. So just a huge amount of extension growth, root length growth, as a consequence of that root pruning treatment. So again, we had intended to measure root length as a response variable, but having spent roughly half a day to try to get this one root out of the ground, we decided to abandon that fairly quickly. The other thing worth mentioning is that obviously as those roots got smaller and finer, the air spade is a rather well, it can be a rather brutal tool for trying to exhume very, very fine roots, and so we were noticing that we were damaging those particularly fine roots as well. So for a variety of reasons, we abandoned that root length as a response variable. So just a couple of images before I pass it over to Andrew to go over some of the more quantitative results. But just a couple of images of some of the types of regrowth that we found. So this is an image that you've seen before. This is that alien sort of face sucker type response. Relatively large roots regrowing from the callus tissue adjacent to that cut face. The next photo shows, again, that more typical response, that sort of beard of regrowth, lots of relatively fine root regrowth from the callus tissue. And the thing that I point out here, well, a couple of things that I point out here is a lot of the root regrowth that we saw happened relatively close to the cut face. Not all root regrowth was that way, but the vast majority was. And then maybe just as a point of interest, there's a root that sort of goes from top left to bottom right, over top of, Andrew's got the pointer on it there. It goes over top of the root that we were actually interested in. And that was just another root from the same tree that had fused to the top of the root of interest. So again, just sort of showing that characteristic that some species do have this sort of root fusing ability. Final couple of slides before I hand it over to Andrew. This just shows a couple of results for the discoloration or the decay that we looked at. And it also allows me to tell you how we measured that cross-sectional area. So what we had here is we took high resolution images of these bisected roots. The ruler that you see in those images allows us to scale the image, so we can essentially use software, in particular a piece of software called ImageJ. ImageJ might be familiar to some of you. It's essentially just image processing software that allows you to undertake measurements as well. So using the ruler as scale, we were able to get an estimate, or fairly good estimate, of the square centimeters of discoloration present on each root. It's not an automated process, I'll point out. Andrew spent countless hours tracing around those areas of discoloration with a mouse. So a huge amount of work went into that as well. So what you see, I guess, at left is sort of a palish discoloration, but more, I guess, consistently, we saw sort of a darker brown discoloration in the roots, and that's shown in the image at middle and the image at right. All of these are our junction treatments, and the next slide simply shows the same thing, but for our internode treatments on this slide. So this is our internode treatment. So same sort of deal. ImageJ used to measure discoloration area, and the area in dark brown there represents those discolored areas. So that's all the sort of anecdotal set of results and the methods. And now I'll pass it over to Andrew, who will basically take up the mantle and talk to you about the quantitative results.
All right. Well, thanks, Justin. Yeah. Okay. Let's have a look at some numbers. So we're going to begin. These are the roots that we cut. I think Justin explained earlier where the intention was certainly not to cause any harm to these trees. They were public trees, as you would have seen in the photograph. There were a number of occasions when we actually uncovered some quite substantial roots under the ground, and we just covered those up and walked away. So what we're seeing here is that the great majority of the roots that we were cutting are sort of between 30 and 50 millimeters. So I guess 1 to 2 inches thereabouts, with a few smaller ones, and then maybe just 1 or 2 slightly larger ones as well. We wanted to get a nice range of sizes to include in the study. 2072 was the total number of new roots that regrow. Justin explained earlier that the root measuring process was quite a laborious exercise. Certainly for me, it seemed like orders of magnitude more than 2072, but that's what the data says. And this is what they look like. So these are the new root diameters. This is how big the new roots were. And again, what we're seeing here with the box is that most of these roots, these new roots, are sort of between sort of 1 and 2 to 3 millimeters. So, you know, predominantly still quite small, fine, that beardy kind of growth that Justin was describing, and often non-lignified. But again, up here, some slightly larger ones, perhaps some of these bigger ones belonging to that face-sucking root that we saw earlier. This is what the root diameter looked like across each of the treatments. So this color coding that you see here will be consistent, followed through the rest of the figures that I show you. So in pink on the left, this is just everything, all of the data sort of bundled into one. The green is the internode treatment versus the junction treatment. In blue is the seasonal effect, which is spring versus autumn. And in purple on the right-hand side is what we call the interaction term, when the two treatments are combined. So again, similar to the previous one, again, most of these roots sort of around that sort of 1 to 2-ish millimeters. And what we're seeing here is that there was no significant effect of either of the pruning treatments on the diameter of the root. So for me, at least, that's quite interesting, that it didn't really matter how and when we pruned these roots. The diameter of the regrowth was sort of consistent across each of the treatments. So this is the number. Remember, we counted all these new roots as well. So this is the average number of new roots per treatment. Again, the color coding is the same. Now we have a few trends here, but because these letters are the same, we wouldn't necessarily report these trends as a significant difference. This one here we would. So what we're seeing here is that the internode roots, the ones that we cut to stubs, grew significantly more new roots than the ones that we sort of reduced the junction treatment. So in that sense, perhaps behaving very much the same way as a branch might behave if we were to contrast these two types of pruning in the crown. Now, we've been able to, with the photographs that we had, we've been able to present some of these results, some of the data we've been able to put in photograph form, just sort of to help you interpret that. Not all of them, but this one we have. So on the right-hand side, this root that you see circled in green, that was the original sort of side root that we pruned back to. Everything else is regrowth. So very clearly, you'll see there that the internode root, the one we cut to a stub, significantly more, substantially more new roots than this one that we reduced back to. Justin described a process where there was a photograph of me and I had the caliper and I was kind of measuring along the roots. So as we were sort of recording the position of each of those new roots, we divided the original parent root, the one that we pruned, we divided that up into these sort of 10 millimeter bands. So 0 to 10 is within 10 millimeters of the pruning cut, 10 to 20, and so on. And by measuring the position of each of these new roots, we were able to place it into one of these 10 millimeter bands. So what we're seeing here very, very clearly, and threefold order of magnitude difference here, is that most of those new roots grew within 10 millimeters of the pruning cut. So again, behaving sort of quite similarly or almost identically to the way a branch might behave if we pruned it. A lot of those new roots emerging from that sort of zone of trauma and the exposed differentiating tissue, dividing tissue at the cambium. Again, I've got this one into photographs. This one, if you'll appreciate, I'm sort of just tilting that root away from me. There's a lot of soil on there. It's difficult to, but the pruning cut is sort of here, and all of these roots kind of growing from the end. Same on the right-hand side, obviously far fewer new roots, but you'll appreciate all sort of emerging very close to the site of the pruning wound. So this is the cross-sectional area, the regrowth percent. Now you can think of this as a sort of a crude proxy for recovered hydraulic capacity. So we measure the cross-sectional area as Justin described, and when we show that as a percentage of the original root's cross-sectional area, it gives us a bit of an idea of how much of that root's ability to get water from the soil has returned. So again, what we're seeing here in green, in the sense that the internode roots grew more new roots than the junction roots, they also recovered more of the parent root's cross-sectional area. There could be two reasons for this, neither of which we investigated, so we're just sort of hypothesizing here. The first reason possibly is when we make that sort of reduction cut, the junction root, the side root that we leave behind, it does a bit more work. It's going to increase hydraulic activity to make up for lost hydraulic capacity. It basically gets more water from the soil. This internode root doesn't have a friend nearby, so the only way that internode root is going to get more water is by recovering more of that hydraulic capacity and growing more roots. The second reason, which again we're just hypothesizing, there could be an upstream influence of hormones. So there's all sorts of hormones, cytokinins, and all other things floating around the root system. So if there's still sort of an upstream influence of hormones, when they make it downstream to the pruning site, they could be having a suppressing effect on root growth. Again, just sort of hypothesizing at the moment. Okay, this is quite interesting. Again, we're looking at the CSA regrowth percent, so it's that sort of proxy for the recovered hydraulic capacity. And the ones I want to draw your attention to is this one in the center top, middle right, and bottom left here. So you recall Justin described we divided these roots into 4 quadrants. So 1, 2, 3, and 4, with 1 and 4 in the upper half of the root, 2 and 3 in the lower half of the root. What you're seeing here, these images are, the figure is scaled. The numbers are not shown. The numbers aren't important. It's the scaling here that's important. If you imagine this as a sort of a slice, as a pie, so the bigger each slice of the pie, the greater the recovered cross-sectional area of root growth we're seeing. So what we see, these are the 3 internode treatments. This is just all the internode spring and internode autumn. And what we're seeing here quite clearly is that those roots are sort of preferentially allocating that CSA regrowth percent in the lower half of the root. Again, we're hypothesizing, but very much probably this is due to sort of more favorable conditions lower down in the soil, wetter, moister. It's just a nicer place for roots to grow low down. This one's the decay index, and here is the interesting difference that we're going to talk about this morning or this afternoon. And what we're seeing here is that roots pruned in spring had significantly more, or proportionally, relatively speaking, more internal discoloration than the roots that we pruned in autumn. Again, 2 possible reasons for this. One, it does get very, very cold in Christchurch during winter. Frost on the ground is very common. Snow, certainly not out of the question. Those cold temperatures could have a sort of a slowdown effect on the soil sort of fauna, the microbiology, the fungi in the soil, just maybe slowing things down. And whereas in spring, everything's kind of warm and wet, and those fungi have a bit of a head start. The other possible reason for this could just be seasonal carbohydrate flux. Platanus, of course, are a deciduous tree, so as we get into autumn, heading into winter, when we prune these roots, the tree is mobilizing those carbohydrates in the crown, sending them down to the root system, and basically providing an immediately accessible sort of carbohydrate resource for that codic response. Whereas in spring, those carbohydrates are going in the opposite direction, and maybe we've just not done the trees too many favors by sort of pruning here whilst that resource is being depleted. Again, we didn't investigate this specifically. We did talk about some iodine staining and some chromatography, but it came down to time and resources, but possible reasons why. So this one again in photographs, I'm showing you opposite ends of the spectrum here. So it's a dichotomy just so you can appreciate. So 2 internode roots, 1 pruned in spring, lots and lots of discoloration here, you'll appreciate. And then the one that we pruned in autumn, only just that little bit of brown here around the tip. Okay, so this is aspect ratio. So we briefly talked about aspect ratio earlier when we talked about branches. We also looked at aspect ratio with the roots as well, just with the junction roots anyway. Non-significant results. So this is aspect ratio versus the decay index. If you're familiar with a paper written by Ed Gilman and Jason Grabowski from early 2000s, I think, they looked at aspect ratio when they pruned oak trees. Again, the same way that we pruned roots. And you can use aspect ratio, or certainly the paper that Ed and Jason produced, to sort of inform your pruning practices. And they had a non-significant result. And we had a very similar range. So aspect ratio of 0.3 to around about 1.8. And which is pretty much the same as what Ed and Jason got as well. So non-significant, it's, to me at least, that's significant, that we don't need to think about aspect ratio when we prune roots, when we think about how that might affect the internal discoloration or the dysfunction of that root, at least within this range. Okay, so wrapping up, let's have a look at some conclusions. These are the research questions and with the answers now. Do tree roots respond differently when they are severed at different locations? Yes, they do. Roots pruned internodally grew more new roots and recovered more of the parent root's cross-sectional area than the roots pruned to a side root. And do tree roots respond differently when they are severed before or after an active growing season? Yes, they do. Roots pruned in autumn had less discoloration than roots pruned in spring. Okay, so here is your hopefully useful take-home message. Where possible, which I will introduce as a caveat, I accept it is not always possible to coordinate the timing of your root pruning, but where possible, root pruning should be undertaken in autumn to help minimize the area of dysfunctional wood inside the pruned roots. And to minimize new root regrowth following root pruning, roots should be pruned back to a side root, like a branch reduction cut. So when might you want to minimize that root regrowth? Well, perhaps a tree has got roots infiltrating a sewer pipe or any kind of buried utility pipe. Then you need to go in there and excavate that pipe and cut those roots and clear that pipe. You don't want that tree to put roots back into that pipe. So you would make a root reduction cut, which will help to minimize that regrowth and minimize the potential for those roots to re-infiltrate the pipe. To maximize new root regrowth following root pruning, prune internodally, leaving a stub. When might you want to maximize that root regrowth? Tree transplanting for one, or perhaps you're doing some emergency work or just some kind of temporary work under the ground and you need to prune some roots to be able to facilitate that. But once you've finished, you want that tree to make a swift recovery as quickly and as well as possible. So you would backfill with some good soil and prune those roots to stubs to help promote as much regrowth as possible. Thank you to our supporters, of course, the Tree Fund, our hosts today. Very big thank you to the Tree Fund for supporting this work. Must also acknowledge Christchurch City Council for allowing us to essentially destroy Hagley Park on 4 separate occasions, I think, looking for roots. And they also provided a very willing and supportive team of people to help us clean up. And the New Zealand Arboricultural Association as well, who provided some funding for the project. Now, I mentioned that we just published this. This is hot off the press, I think just 2 or 3 weeks old. That's the bibliography there. Hopefully that QR code should take you, fingers crossed, to a free download link, hopefully. But yeah, that's us. Thank you.
Thank you, Dr. Benson, Dr. Morgenroth. Fantastic presentation. I just very applied research with some very clear results. So thank you all for this, and thanks for taking the time to present today. We have a lot of questions. I'm not sure if we'll get to all of them, so let me just go through a few of these. There was one question here. It says, what was the largest diameter root you cut? And did you find differences in regrowth response, better or worse, based on diameter size?
The largest diameter. So Justin sort of alluded to this when he was describing the methods. Rarely did we see concentric roots. They were all sort of all over the place. So our measure of diameter has been reverse engineered from an accurate measure of cross-sectional area. But when we did it that way, it was just over 100 millimeters, I think. Did we see more regrowth with increasing diameter? Off the top of my head, I don't remember. We will have that information, but logically, yes, because there is a larger exposed region of traumatic cambium from which to grow new roots. So yeah, the answer would be yes.
Thank you. Another question here. Did you all have a chance to do any culture? Somebody asked if there was any culturing of the decay organisms found at cut-ins to count frequency of which were occurring so that you could understand which was causing the most decay. Did you all have a chance to look at that?
I can answer that one because it's an easy one. The easy answer is no, we didn't. It's a great idea. It's a really interesting question, but we were somewhat limited, I guess, in what we were able to do given the funding and time that we had. So as generous as the tree fund was, once you start getting into sort of chemistry tests or lab tests like that, the money disappears fairly quickly.
Understood. And there was a number of questions related to, what drove you to pick the species you chose, and also, maybe replicating the study with other species or other soil types as well came up a couple of times.
I can say that if you want, or do you want to?
I mean,
Yeah.
I guess I would simply say that all those suggestions for other types of research are amazing. Please keep them coming. I think there's no lack of really good ideas in the chat and also in the areas of the questions. So I think those are wonderful ideas. Unfortunately, we weren't able to do all of those things. Andrew and I continue to talk about the types of research that we can continue to work on together because we both do have this sort of shared passion for tree roots. In terms of why we chose the plane tree, a lot of it was I guess it was a combination of a pragmatic decision. Those were the trees that were available to us, but also perhaps in hindsight, it was probably a good idea to use quite a hardy species, a species that can tolerate a little bit of abuse. Obviously, we worked in a very public area. We worked with older trees. Had we been working with a species that was maybe more sensitive to root damage, we might have created a situation where we damaged or even killed some of those trees. That's certainly not the case years later. They're in great shape. So I think maybe in hindsight, it was quite a fortuitous choice to choose such a hardy species. The other thing that goes along with that is because we did choose such a tough species, they were able to exhibit quite a lot of response that we were able to measure. I don't know whether you wanted to add to that, Andrew.
Yeah, I just have a couple of things. I mean, Platanus is sort of the universal tree species. It's this is applied research, and we wanted to make it applicable or useful to as many people as possible. And I think most places in the world have got Platanus trees, so that was kind of one of the reasons. Again, just to add to Justin's comment that we were playing with public trees, and causing too many problems wasn't going to be a great look for us. But originally, when we embarked on this with wild ideas of including, I think, 3 species, we had some birch trees and some pine trees as well, sort of earmarked for this. And it was really only when we started working with the air spade and looking and severing roots that we really got an appreciation for just how time consuming that was. And it was, I think initially we were going to do maybe 20 replicates of 3 species. But when we realized how much hard work and how time consuming it was, we just made a decision to do 1 species with more replicates. So yeah, we did think about it, but it went in a too hard basket pretty quickly.
Yeah, I mean, in the end, I think just to help people understand sort of the scale of that air spading work, there was 4 separate periods. There was 2 periods of 2 week-long periods of I guess air spade work to allow us to prune the roots, and then 2 subsequent week-long periods of air spading work to expose those roots. So we were on the end of an air spade, or rather on the end of 2 air spades working side by side for the better part of a month over the course of the 3-year experiment. So it's pretty easy to understand why I hope it's easy to understand why we sort of limited it to just the single species in this case.
No, very understandable. I also see there's a number of questions, I think very good questions, looking at, the correlation to tree stability with root pruning, which I don't think you all had a chance to look at that either. But I think it's also a fascinating, how much can you prune before the tree you start losing stability? And so do y'all have any comments to that, whether from other literature that you can point people towards?
I mean, there is lots of good research on that. It's hard to recall any of the specifics off the top of my head, but I'd probably just point people to the BMP on working with, or I guess the BMP on working with roots, and that's a good starting point for some of those detailed answers.
Alessio Fini did a really interesting study a few years ago. He looked at growth physiology and stability responses in 2 or maybe 3 species. I think that was published in 2020, but he's got some really interesting stuff on tree stability from pull tests. Yeah.
Well, thank you all to both of you. We are approaching the end of our hour. Thank you for a fantastic presentation. Thanks to all of y'all who participated. And I'll just remind you, we'll be back in December, December 9th, with another presentation. You can go to treefund.org. You can find more about that information. And just a quick reminder of CEUs, there's nothing any of y'all need to do. When you registered for this webinar, you gave us your name, you gave us your certification number. Heath Hupke is going to compile all that information, send it to ISA on your behalf. Give ISA some time. It's a lot of data. I see there was, I think, almost 1,300 people on here at one time, which is fantastic. And just as a closing note, thank you, Paul Putnam and Tree Fund for the kind words and the recognition with the Ken Ottman Award. It is very much appreciated. As I noted in my comment, this webinar is a pleasure to host and moderate. As you can see with the 2 wonderful speakers that we had here today, coming from across the world, from way deep south, sharing new and emerging research just published 2 weeks ago. So thank you to everybody. Thank you to Tree Fund. Thank you to Dr. Benson and Dr. Morgenroth. And see y'all back in December. Cheers all.